Good evening. Good evening. Check 1 2. Microphone check 1 2. Check 1 2. Good evening, everybody. Hello. Welcome. Hello. How's the speakers? Here we go. We're getting there. I'm actually very excited to be standing up here tonight. Good evening, everybody. My name is Jackson Hudecki. I'm the incoming president on the board of the Hamilton Naturalist Club. I'm currently the vice president. So next month, things change over and we take on a bit of a new board. New similar board, but regardless, I'm excited to have everyone here tonight because this officially kicks off our 2025-2026 series of speakers with a very interesting talk, which we'll get to in a few moments. But a show of hands, is this anyone's first time coming to a monthly meeting? Awesome, welcome. So the Hamilton Naturalist Club hosts a monthly meeting from September through April, and we also host the week after a bird study group meeting. So where these talks can be very nature generalist, the bird study group is very bird heavy. So if you're a birder or just a bird fan in general, or just a nature nerd, you have an option to come to 2 meetings a month if you wish, and they're gonna be here right through until April. So excited to have you here tonight. Raise your hand if you haven't been in a while and you're back for the first time in a bit. Awesome. So welcome back. And if you're a fan of nature in the city of Hamilton or in the region and you're looking to support, lots of ways of doing so, and I'll chat about that in just a couple of minutes. But in case you need a reminder, these nights typically last from 7 until 9. 7 to 7:30 is a bit of an open half an hour, a mingling time where we have refreshments thanks to Dina and Ellen. So quick round of applause for Dina and Ellen. So feel free to help yourself to cookies and a drink. And then at 7:30, the official meeting begins, and it's usually just some announcements, just some reminders, things that are going on with the club before we introduce our speaker. That lasts for anywhere from half an hour to 45 minutes, plus a Q&A. So feel free to think of some questions throughout the presenter's talk. And if this is your first time and you want to support nature in Hamilton, consider becoming a member of the Hamilton Naturalist Club. Or perhaps you're ready to take on another step, and it's to become a volunteer. Lots of ways of getting involved with the HNC. We often are looking for anyone who's a photographer or a writer who wants to submit something to our monthly publication. One of the perks of being a member is you get a monthly magazine mailed to you, and we've got a great team of folks who are willing to entertain any sort of subject that you send them. So if there's something going on in your world you want people to know about, something you're excited about, something you want to share, feel free to send it to our Wood Duck team. It's just woodduck@hamiltonnature.org. And it's a really neat way, again, to just kind of create and join that community. A neat thing that's starting shortly is something called Sanctuary Stewards. So the Naturalist Club oversees 11 nature sanctuaries in the region. And we're looking for some folks who would be interested in going to and, again, stewarding one particular sanctuary that you would then report back to the sanctuary committee about what is happening at that specific location. It's a really neat project, something that we are spearheading. So if you would like to learn more, you could contact Jen Baker, who is here but is leaving shortly, land@hamiltonnature.org. Or check out our website for more information. But it's a great way to take your skills, and they could be minimal, in order to just let us know what's going on at these sanctuaries. So that would be great. We have a lot of events coming, a lot of ways of getting involved. So on our website and on our social media, there's a way to see all of this information. So oftentimes while we are stewarding sanctuaries, invasive species, if you've ever worked with invasives before, can get a little bit out of hand. And so we are always asking the public for some help with invasive species pulls. And this one in particular is going to be at the Sheila Dunduli Nature Sanctuary, which the timing of this is cool, 6 p.m. to 7:30. And I only say it's going to be cool because it gets dark pretty early now. And so you might encounter some... Yeah, so you might get a chance to see some cool nocturnal wildlife while you're out there helping. But if the evening is not your forte, there's also a morning one, but a really amazing sanctuary and not too far from here, and named after a really cool person, if I might add. Tree plantings are happening on October 8th and the 18th. So if you wanted to put your muscles to good work by planting trees instead of just ripping some out of the ground, we could use your help there. And there is the Cartwright Nature Sanctuary Tour, hosted by one of the gentlemen who's on our sanctuary committee, which is great. So that's on, again, an evening walk. And the Cartwright Nature Sanctuary is just gorgeous, and it gives you a really cool view of the region if you haven't seen it. So that's on the 17th of September, which is tomorrow, or 2 nights from now, sorry. The Grimsby Wetlands Tour on October 15th. It'll be hosted by Bruce and Lori McKenzie. So these two folks have been stewarding this particular sanctuary for many years, and it really is just a gem of a space. If you've never been, consider going to that just to hear from these two folks who've turned it from like a sewage lagoon into a very bustling nature sanctuary. There's a birding event on October 8th with Barry Coombs at Bayfront Park. Mycelial Mysteries, a spooky fall fungi foray with Blair. So if you attended the last fungi outing, Blair was awesome. So they'll be doing something else on October 11th from noon until 2. And the Nature at McMaster Community Bio Blitz. So that is the 27th of September. Registration is required for that, but this is cool because we're asking folks to come out and honestly just take pictures of and point at nature and saying like, what is this? And that way we can capture a little bit about what types of communities are living among that sanctuary at the McMaster Forest. And then I hope we have enough plants for this, honestly, because I've been seeing this promoted everywhere. The Native Plant Fair on October 4th, it's going to be in this parking lot. Yeah, I've been seeing a lot of people talk about this. So if you are interested in coming, come early. And if you have plants, I mean, I don't know if we're taking plants, probably not. Is anyone here, is anyone on our team running this who's here right now?
Would you like to know?
Well, are you going to have enough plants, do you think? Do you need plants?
We will have plenty.
Plenty, okay, good.
I've been in touch with all the growers.
Perfect.
They're bringing trailers attached to their trucks.
Okay, good.
Asking for 3 tables a piece. So I don't think there will be a problem.
Great. Forget I said anything. Come buy lots of plants, put them in your gardens. Let's increase biodiversity in Hamilton. Thank you, Jean. Okay, and as mentioned, the bird study group meeting, our first one of the year, will be on Monday coming up right here with George. So George has been a member of the Hawk Cliff Raptor Banders, and we'll have a wealth of knowledge and some great photographs. So celebrating 50 years of Hawk Cliff Raptor Banders, that will be the subject next week. So hopefully you'll come back for that. And in 1 month from now, it will be the club's AGM, so that's October 20th, right back here. And so while this is an AGM, it also is a chance for the member base and the community at large to learn about what is happening with the HNC. We give reports about a bunch of different topics, so it's a chance for you to come out and learn about all that we are doing. So that will be next month. And so we'll have Kaja from the Natural Areas Inventory providing an update, as well as staff providing an update about our sanctuaries. So that will be on the 20th, right back here. Okay, enough from me. So our special guest, Alice Dabrowski from the University of Guelph, is a Ph.D. student whose research documents flower fly larval diversity and ecology in the eastern regions of Turtle Island. Her work aims to produce field guides for community science projects and develop low-cost flower fly larva options for aphid control in remote greenhouses. Beyond flower flies, Alice has a background in restoration ecology and environmental science. Everyone, let's put our hands together for Alice.
worries. Hi, everyone. This is my first meeting. I've never come to a monthly meeting here with the Hamilton Naturalist Club, but there were already friendly faces in the crowd, I'm sure online as well. But it turned my nervousness into excitement like very, very quickly. So thank you so much for being so welcoming and here to listen to me talk about probably the coolest fly out there, the coolest maggots out there. Hopefully I'll be able to convince you of that by the end. And I will just switch over to my presentation. Perfect. All right. Quick, simple, easy. No technical difficulties. So today's topic, I'm going to be discussing Ontario flower flies. I am a Ph.D. student at the University of Guelph School of Environmental Sciences, and I'm co-advised by Dr. Andrew Young and Dr. Jessie Popp. Dr. Andrew Young is actually here, so if you'd like to talk to us later, or if you have anything good to say about me, he is present. It wouldn't hurt. See. All right, so before we dive into the technicalities, a bit about who I am, kind of where I come from, and what values I try to uphold in the work that I do. So my journey started off at U of T doing political science and environmental ethics, so something, very distant from what I'm currently doing. But that's where the journey started. After my undergraduate degree, I volunteered at the Royal Ontario Museum doing things like outreach for environmental awareness or environmental topics, just getting to talk to people. And that's where it really ignited this passion in me to learn more about the world that I'm living in and the natural environment around me. So from there, lacking in some of the field skills of a political science degree, I went to Niagara College for their ecosystem restoration program, and I loved it. It was just before COVID, and that experience showed me that I was ready to commit far more time into learning more about ecosystems, specifically, plant communities, and what things like to live together, what makes a healthy ecosystem, and how can we best support that. So I've done my master's at the University of Guelph. It was a course-based master's. And this kind of leads into my next point, but I had the opportunity to take a field course in entomology in Costa Rica with Dr. Young. And as somebody who comes from a very strong interest in plant communities, I was thinking, plants, well, insects are important for plants. It wouldn't hurt to know a little bit about insects and familiarize myself with, the things that are eating, the things that are pollinating, the plants that I love so much. And that really changed the trajectory of my life. I didn't expect to like insects as much as I do. There's a specific group, I will say, but it really has changed my life in ways I didn't anticipate when I first took the course. If anyone's watched Alice in Wonderland, falling down the rabbit hole, that's exactly what it felt like. Curiosity leads to trouble. And for me, that looked like taking on a Ph.D. role, committing another 4 years to a group of flies. And throughout all of these experiences, I would say that my values tend to revolve around finding and honoring connections, slowing things down, going outside, noticing what interacts with each other, and doing what we can to support that. And as well as, the human side of things, which is empowering and engaging people like you guys to take their passions and what they love out into the world and bring us closer together and working on something together. So it kind of leads into, yes, enthusiasts like you. I hope you become as enthusiastic as I am about flower flies by the end. But we can start off in kind of this broader thinking about insects because, I've heard from old and young, people will see an insect and they'll be like, gross. Or, the unfortunate thing of like, how can I kill these wasps that are pestersome and bothering me? And it's important to think about insects playing a role in a healthy environment. So we might not think of this all the time, but they do play a crucial role in things like energy transfer, with the everyday things that they do, the everyday services that they provide us. So things like decomposition, population mediation, there's pollination, food source for things higher up in the trophic levels, and seed dispersal. And because these are happening at such a small scale, you don't really pay attention unless you slow down and you take that time to appreciate it. Because really, we might be, these very dominant creatures, but without insects, there would be nothing for us, right? We really do rely on each other. So there are a lot of insects out there, an impossible number of insects if you're looking even at the species level. But I have found myself enmeshed with a group known as syrphids, or flower flies, or hoverflies. And this is kind of what we're going to be looking at today. Now, kind of show of hands, who is familiar with flower flies or hoverflies? What? Oh my gosh! I don't usually get that many hands. Okay, well, who likes maggots? Okay, we have somebody doing a little heart. Okay, I'll take it. We'll start off with the adults. So believe it or not every single black and white thing that is visiting your garden or visiting a flower is a bee. Flower flies can be, not all of them are, but some of them can be very charismatic, very convincing mimics. So, it's a fly trying to be a bee, so it's a wannabe. Oh, I've been waiting to use that one. Yeah, right there, that little buzzy thing, it's a pixel, or like it's, animated graphics, but that's the idea of a flower fly. And technically, if you're looking into it from a taxonomic perspective, they're flies in the family Syrphidae, so that's like their scientific name. Many of them are charismatic, they're known for their hovering behaviors, which is kind of giving you an idea of the reason behind their other namesake in Europe, which is hoverflies. And despite them sometimes being very convincing, having those physical and behavioral mimicry, they can't sting or bite you at all. There's actually nothing about their bodies that can hurt you in any way unless they flew into your eyes or they bombarded you. That is the only way that they could even remotely hurt you. So it makes a really fun outreach model of insects. People warm up to something if they know for a fact that they can't be hurt by it. So adults, they have these sponging mouth parts, like many other flies, that they'll use to feed on nectar and feed on pollen at the flowers, as you can probably guess. And what's interesting compared to compared to bees is that, like mentioned here, flower flies are non-provisioning. So who knows I saw that you guys had a bee talk a few months ago, or maybe a year ago. Yes? Was anyone there? Okay. Well, something that's interesting about bees is that their brood act as this center place in which they'll go out and forage, and they're kind of limited in distance based on where that brood is. Flies, they don't have that kind of parental care, and which makes them available to kind of go across the landscapes and really investigate and follow those floral resources wherever they are. So their travel across a landscape can be substantial, and in some cases, I have a colleague, a fellow student in the Young Lab, who's actually written a review paper on syrphid migration specifically, so flower fly migration. Everything you associate with monarchs, this magic ability to cross an entire continent. For some flower flies around the world, they can do pretty much the same thing, and that is incredible if you think about their size. Very, very small. Okay. So they're incredible pollinators, given their association with flowers, the fact that many of them do have quite a bit of hairiness to them, which helps the pollen stick. And the migration allows for that pollen to transfer across a very long distance. And that helps with the genetics of plants and flowers and the things that we can come to appreciate. So in our region, many of them would be considered generalists, meaning that they don't have a strong association with, any particular flower, which is great because it means that in urban centers like Hamilton, or places where we have reduced access to natural areas or, those pristine areas, flower flies can still manage to thrive. And that's really impressive, considering like how many things end up declining in their populations or really, really struggling when we introduce human activity. All right, and on to the maggots, the larva. Woo. Can anyone spot it? It is there.
Oh, there it is.
All right, I tried to make it easy. I'm glad people picked up on it. So that's exactly right, it's right there. And the immature stages, the larvae, the maggots, you can kind of refer to them in all these different ways, but they lack legs, they don't have eyes, and they don't have a head capsule, which would be what you kind of associate with caterpillars. I get a lot of people asking me like, oh, what's this thing? It kind of looks like a caterpillar. Well, if it's got a head capsule or, something that looks like a helmet, it's not a caterpillar. Maybe it's a flower fly. So the larvae can actually occupy different habitats and different ecological niches. So I have a syrphid larva or a flower fly larva on a plant here, but actually they can take on different feeding modes. So they can the larva can feed on bacteria and, what was mentioned earlier was like a sewer pool, right? Like there are some flower flies that might actually love that. So they're very resilient, very durable in some cases. But what's interesting is that of the 4 subfamilies, 3 of them are predaceous. So these eyeless, legless, headless maggots are somehow able to find their prey and, hunt things down. And that is so impressive. Like, think about how difficult that would be. Oh my gosh. And then on top of that, think about how they're operating at their small size in a 3D space, right? Like it's not just a side scroll game that, you can jump over things and it's fairly 2D. It's a 3D space that they're able to navigate in order to continue their populations. All right, so as you can probably guess, you don't get very far when you're eyeless and legless. So larvae tend to be restricted to a very small area for that part of their life. And if you can guess, in a scientific perspective, it makes them very difficult to track, right? So nowadays, scientists have passive trapping methods like malaise traps or pan traps. They're traps that you can leave outside and not really look at. You can visit it once in a while, collect your sample, but for the most part, like you're not putting in the work to find these things. So these larvae, unless you have the trap right up against them, they're not gonna somehow find their way into the trap. It's very unlikely, making it difficult to track and study populations at this particular stage, right? Oop. And kind of going back to, bacterial feeding. One thing I will say is like, oh, that's kind of gross. Like, how do we benefit from flower flies that feed on bacteria? Like, ugh. Like maybe let's convert habitat into something else. But I will say that flower flies or the larvae that are feeding on the bacteria, they're making that energy accessible to other areas of the food chain. So think about things like fish, birds, turtles. They're not necessarily going to be eating that kind of mucky, disgusting substance. It's the flower flies that make it available for other beings. And then the predaceous side of things, the predaceous larvae are interesting as well because their ecosystem services or their ability to feed on kind of like soft-bodied insects like aphids, right? Who has a garden? Yeah, who's seen aphids? Yeah. Now, Have you noticed a flower fly before? Yeah? Okay. Because I will say that oftentimes if you have a very strong colony, you will see at least one. It's probably hiding, so you have to have very keen eyes. But thinking about that, taking it out of your garden and putting it into, let's say, an agricultural setting, right? Farmers have this whole big operation. Oftentimes it's a monocrop, and it can cost a lot of money to put pesticides on their crops or manage for, certain pests. And flower fly larvae, if they're in the area and there are good enough populations and they're able to find these aphids, they can actually contribute a significant amount to biotic control, so that naturally occurring benefit, without farmers necessarily doing all that much, without too much of a cost. So that's really impressive, and I would say that's kind of what's leading the science side of things at the moment. It's the fact that there's an economic kind of tie-in for farmers. And Ontario, you drive down to the south, you're going to be seeing crops. So you can imagine how impactful that can be for the people who are leading those operations, or their livelihood depends on it. So studying them, very important, but very tricky. That is kind of the point. Sorry, one sec. Okay, so I've tried to paint this picture of, why the adults, why the larvae of these flower flies are important, why they would be interesting maybe. Well, how would you be able to go out and identify flower flies? Well, we'll start with the adults because they're probably the easiest to spot. And I wouldn't recommend catching it with your hand right off the bat. These often look like bees. Make sure it's a fly first. Make sure it has 2 wings, halteres, which are these almost like golf club-like appendages. Oh. Okay. It got back up very quickly, so that's good. But they have these halteres, and that makes it a fly. All wasps are wasps or bees or Hymenoptera. Many of them are going to be having 2 pairs of wings. And in addition to that, I wouldn't say that this is always the case, but you can look for a short antenna. If you're able to look at its head and get a clear view of its head, if it has longer antenna and, the 4 wings, I'm gonna guess that it's a wasp or a bee. But the short antenna is what tells you that something is suspicious. So this first pair of characters, that's going to get you to a fly. Well, how do we know that it's a flower fly? So at this point, you would be looking at the wings. If you have a loop or like a lens or even a very good camera, that can typically do the job. But what you're looking for on the wing is in the center of the wing. It's that little vein right there, and that's known as the spurious vein because it starts off kind of right there, and then it just trails off. It doesn't connect at the other end to any of the other cells, or the other veins rather. And there are some other fly groups that have this, but for the most part it is associated with flower flies. All right, and then from there, it gets far more complicated when you're looking at the family level or species level, but generally you're going to be looking at the presence of a wing spur. That would indicate microdontidae. The mouth shape, if it has a notch in the margin of the mouth, then that would be Aristallinae. If it's a rounded mouth edge, Pepizinae, and then if it has post-pronotum pile, it's a very fancy way of saying does it have a hairy shoulder at the back of the head? And then that would be Surfinae. And the larva. So like I mentioned, they have no head capsule, so like that helmet-looking thing, no eyes and no legs. If you can confirm that, then that is going to be a brachyceran larva. And then from there, you're gonna, again, this is hard because larvae can be quite small, but you're going to be looking for the posterior respiratory process. So you have the head, right? And on the other end, you're gonna have these tube-like structures that they use to breathe. In other fly larvae, they'll be separated either very obviously or, sometimes not so obviously. But take a good look. If they're fused, then that's going to be a syrphid larva or a flower fly larva. And then if you're to go further than that, again, this is getting into the nitty-gritty of things. Consider things like the habitat and what it looks like. For the most part, so for example, this is Aristallinae. Has anyone seen this before? This is actually pulled from iNaturalist observations in Ontario, maybe even Hamilton. But have people seen these? I find that these often are some of the most reported. Well, so they're commonly known as rat tail maggots. I don't like that name because it's a maggot, fine, but that's not a tail, that's more of a snorkel, right? So like I was saying, the larvae, this group is particularly known for elongated ones. They can have varying length and they can live in so many habitats. It's really difficult to kind of pinpoint one specific thing that attributes it to Aristallinae. Then we have Microdontinae. These are myrmecophiles. Does anyone know what that might mean? Sorry, someone said it? Ants, yes. So these are ant associates, meaning that they live all of their larval stage in an ant nest looking like a turtle. And, it's a very interesting shape compared to some of the other subfamilies, but I will say that it has a purpose. This odd shape is actually for defensive measures against the ants that they live with. So there's strategy in it. It's not that they just want to look like a turtle. And then we have Pipizinae. These are not very often reported, even within like scientific collections. And for the most part, it's hard to tell in this photograph, but they're very dull colored. They will have that fused posterior breathing tube, but then on the very end, so like let's say right here and right here, they'll have extended fleshy projections. I thought about this for a long time. I really have no idea at the moment how to explain it better. It's essentially, fleshy projections. And then of course, I didn't get to talk about Syrphinae, which is this example right here. It's a sample that a specimen that we were able to collect. And they are probably the most colorful, I would say. They come in all different shapes, sizes, and patterns. So if you see a larva within, but within proximity to an aphid colony, and it looks really funky, it might be a Syrphinae. Sorry. Okay, so we've learned why they're important. We've learned how to ID them. Well, what do we know about them in Ontario? So taxonomy and applied entomology, using insects for the purpose of these human projects or human activities. It's been responsible for the bulk of the scientific literature and the scientific study of this group in general. So we've had some pretty important resources that have come out of the work, especially, Manual of Neuroarchaeoptera, or this, what is it? It's a PowerPoint key, so very accessible. Both of them free, which is great. It allows people to access the information and take their time if they're looking to learn kind of a bit, deeper knowledge on specific species. Where do I start? I love this guide. I have my copy, which I received maybe in 2022, and it already kind of looks like this. It's beat up. I think I've pressed plants in here, so there might be some pages with stains. I write notes in here, and I have to say, I don't think it's gonna last for the length of my PhD. I use this all the time, and it's what helped me kind of build an understanding of flower flies as well. Like, it really is quite accessible. So for Ontario, based on the work of some pretty incredible people, we have, I think, 303 different species of flower flies, which is, quite a bit, especially because this guide for northeastern North America, I think that has 366 species. So, a large majority of them can be found somewhere in Ontario or have been reported from Ontario before. For Hamilton, I did check this online, and there's so far, hopefully with you guys there might be more, but more than 90 species within Hamilton being reported. And we do have kind of these long-term data sets, which can be really important for things like tracking or population monitoring. But there are parts of the province where work hasn't been done, or it's been in like these kind of blitzes and it's not sustained efforts. So gaps here and there, but for the most part, very well documented in their adult form. But of course, climate change, climate shifts, and new species coming in from far away, that number likely to increase, and new provincial records become more likely, which is, half exciting, half not so exciting. Depends on the story behind it, right? So unfortunately, like I mentioned, flower fly larvae are much harder to find, much harder to track, and there aren't very consistent records of them, I would say, especially for sustained long periods of time. It's impressive, though, if you look at the global scale. There are areas like places, in Europe, where there's a strong community culture around documenting flower flies. So they've really led the way in a lot of our understanding of flower fly taxonomy. But while it's not the same here in North America, I will say that larval interest is growing, which is great. I mean, at least one person, it's me. I'm very into flower fly larvae. But if you look at the numbers and you look at the formal descriptions of things, more than half of the larvae of Surfinae, so those predaceous, really colorful larvae, more than half of them are either undescribed or poorly described. And if you think back to when you're looking to study a group or you're looking to identify what you have in front of you, if you don't have that foundational knowledge or those foundational resources, you're not going to be able to have consistent tracking. So we need a better understanding of the larvae in order to, monitor populations and figure out what we can do to conserve them if that's our goal. And that's kind of where my work comes in. So I don't consider myself an entomologist. I still think I'm very, very fresh, very, very new to this whole thing. But my piece of the work is starting conversations, doing my best to bring people in and build those connections, and maybe get things started on the maggot side of stuff. Yeah, hopefully one day, having a guide all their own. So these maggots, understudied, though scientific interest is building, public interest is building. So okay, now we're going into the nitty-gritty of my work. That was all background. Now it's kind of what I hope to accomplish. So PhDs are typically divided into chapters, and these chapters tell a story. For the most part, they have to be, a coherent piece of work. And for me, that story focuses on blending flower fly taxonomy, flower fly ecology, and also where humans can fit into that as well. And of course, I have a special place in my heart for the larvae. So this chapter that I have up here, it's going to be focusing on developing those formal records. So like I mentioned, more than half of the larvae of this group are undescribed or poorly described, and we have much better technology than what we did, I don't know, in the 1930s, 1950s, right? We have like the things you can do with your camera alone is quite impressive. So just imagine what the microscopes of today are able to capture. So my work on this particular chapter, we have 27 genera that I need to collect at least one species of. And I'll collect these species. I'll try to either rear the larvae or have them sent in for barcoding to confirm their species identification. And then with enough material having built up over time, enough pictures, I hope to kind of find consistent characters that could lead into a dichotomous key. Has anyone done or used a dichotomous key before? Okay, so some people, right? Has anyone done a like a personality quiz in a magazine? Because it's like pretty much the same thing. It's like, do you like cookies or cake? You know, you pick the best option and you move further down the line until you get to a final identification. Hopefully, I'm going to try my best to do species where I can, but for the most part, it's going to be higher level genera. All right. And this work is important because, like I mentioned, it becomes foundational for biological surveys if you're looking at it from like these big project perspectives. But also, if you are looking to generate a life list in your personal life, you need to know what you're looking at, right? So hopefully this will contribute to that. Okay, so in the process of looking for those 27 representatives, this was actually in my first year, so right off the bat, something crazy happened. But it has to do with a specimen that Dr. Young and I collected at the University of Guelph Arboretum. Has anyone been there? Yeah, it's a very nice space. You've got lots of different habitat. I think the history of it is it began being converted or managed as an arboretum in the 70s, right? So not too long ago. But we were able to just kind of look around, see what we can see, start building that collection of material for this project. And we came across a specimen, and we didn't know what we were doing. That's fine, we were just getting started, but we collected it and tried to rear it out. And that's kind of where something became obvious, or it became obvious that something was amiss. Basically, when we rear these larvae into adulthood, we're able to use those resources that I had mentioned earlier to confirm an identification. And when we had reared this particular larva, it became obvious it was part of what's known as a species complex. And that's a fancy way of saying that there are 2 or more related species, but they are so similar to each other morphologically that it's hard to kind of tell them apart. So Eupeotes americanus, which is the larva on the right, and Eupeotes pomus, larva on the left, Eupeotes pomus larva on the right. These are the 2 species that we're already having a difficult time with. They're already part of this species complex. The males, thankfully, have external genitalia characters that make it easy, but the females, indistinguishable from one another. You would need DNA barcoding for that. So this specimen, which I'm still keeping hidden, we reared it out, and the genitalia of the male most closely matched the pomus. Okay, not crazy. But we sent it in for barcoding because pomus is already underrepresented in adult collections, and the barcode actually suggested that it's not most closely related to pomus, it's most closely related to americanus. So physically, it looks like pomus, genetically, americanus. And I don't know if you can tell, but the larvae, very easy to distinguish, right? You know, one's brown, one's green, easy peasy, not a huge mystery. You'd think that if it were either one, it would look like either one. But actually, it looked like that. Completely different. And it's kind of mind-blowing, right? So this is just one example where the story, it tells you that larvae can offer different insight, especially in the areas of taxonomy and even ecology, that it really has gone underappreciated and not explored as much as it probably should have. So adult morphology in this complex is conservative, but you know, the larval stages are what's giving us the first indications that this is something different. And with this particular chapter, I believe, you know, maybe things have changed, but I believe this will be the first case of a new species being described from the larval specimens as well as the adult specimens. Oftentimes, they'll just be described from the adults, and then the larvae come decades later, right? But this is the first time it'll be done with both, as far as I know. And this kind of goes to show, you know, diversity is where you least expect it. This was just in the Arboretum, somewhere I can walk to, like 5 minutes from my office. You don't have to go to the tropics or somewhere that's notoriously biodiverse. You just have to look outside and slow down. Okay. And of course, what's the point of science if it's not accessible to people? Like, why keep it in its sterile little box to be put on a shelf forever and not really looked at until the next unfortunate PhD student comes across that work, right? So for this particular chapter, I'm going to be working with different community groups to co-develop flower fly resources that kind of fit their own research questions or fit their own goals, their own values. And this might not seem necessarily like science, like, you know, I'm not working with hard numbers, I'm not working with models, I'm not working with statistics, I'm working with people. And what's novel about this is the process itself. You know, even today, even in the realm of entomology, even environmental science, science and academics still oftentimes struggle to recognize the importance of, you know, non-experts or non-academics. And they don't appreciate community members as partners in this work. So, you know, kind of a different angle from what has been traditional entomology. Now, when scientific institutions begin to work alongside each other with the public, both groups can benefit. You know, researchers, they'll get additional data points that they can plug into their models and figure out, you know, changes in populations or where populations are shifting. Great. Scientists get their stuff, and then participants actually get to see themselves reflected in the work if they're being consulted, if they're co-developing resources together. And the whole point being that I want to empower community to take on projects and take it in a direction that is meaningful for them. All right, and this kind of work is important. Time and time again throughout this project, I've seen the impact that inclusive science has on generating kind of these really interesting, really new insights into flower flies. And it's a participatory science, right? So that being individuals can contribute equally to a project goals, kind of regardless of their expertise level. And in fact, you might be interested in something completely different from the project at hand. So for example, if there's an entomology project looking at flower flies, bringing in botanists or people who really love their native plants can completely change what the project is able to find by the end, right? So science can really be improved if we choose to collaborate. Now, this project, in this project alone, community science community science participants, they have contributed to things with the undescribed species by simply uploading things. So without looking extensively at collections, I can already say that this undescribed species that we found in southern Ontario actually extends all the way to British Columbia. And that's just because people are curious and they're willing to engage in these community science platforms. To my benefit, like, thank God. Although, I would maybe appreciate going to British Columbia if anyone wants to recommend that to my supervisor. Yeah, and ultimately, okay, like I mentioned before, you don't need to travel to exotic locations or have fancy letters or, 50 years of experience in this field. That can all be great. But really what it takes is an ability to appreciate the natural connections around you and a curiosity. All a person needs to be a scientist is, curiosity and a willingness to learn. So this is kind of a fun ending off point. And it really goes to illustrate the impact of community science close to home. So I'm trying to think how to start this off, but we have that ant-associated flowerfly subfamily that we don't see very often. They're not often reported even as adults, less so as larvae, which we have here. But I was looking at the Hamilton region on iNaturalist, trying to pick up what have people seen around here? What's interesting? Like, what can I talk about that would really connect to the audience? And I came across this observation on iNat, and it was a microdon. So I'm like, okay, that's already pretty cool. And then I kind of realized that I know very little about microdon. That's not the group that I focus on. But I know that the shiny ones can be very interesting. So I don't know if you can tell, but this particular observation, the picture shows almost like this iridescent green to it. It looks like a sweat bee, right? Yeah. So I'm like, okay, I need to dig deeper into this. I need to kind of figure what's going on with this. And I mentioned it to my supervisor, and he confirmed, yes, it's a microdon, cool. And he gave me some suggestions to the species. And then I kind of turned to my handy dandy book to see what people have written about it, or what are the details of this species. And this is what's reported in the book for this particular species. We see a concentration of collections or specimens collected in Florida, but very much a southern species. So I'm like, okay, this book was published in 2019. Maybe people have found additional observations since then that would explain, us seeing this in Hamilton. And no, not really. People have observed it. Which is great, which is really cool, but nothing, the picture is small, I'm so sorry, nothing for Canada, let alone Ontario. And I go on to check some other databases that I have access to, and again, nothing within those databases says that this species has been recorded from Canada. Okay, everyone. That's some crazy stuff right there. And of course, so Mark, who's present here today, he was the one who uploaded this observation, and of course I tackle him down in iNaturalist message, the inbox there, and I'm like, hey, so I think something interesting is going on here, but your accuracy is on the low end, so it suggests all of Hamilton as where you might have seen this. Can you give me some additional information? So he gracefully shares the location of the observation, and just Monday, not this Monday, but the Monday previous, the two of us go out together for 4 hours trying to look for an actual specimen that we can collect. And, we weren't expecting much because it's the end of the season, but no luck, we'll keep trying though. And I hope everyone here also keeps trying, maybe upload what they're able to see. But again, what this goes to show is how valuable curiosity can be, that anyone, anywhere can make some really substantial contributions to science and our understanding of flower flies in general. All right, so to summarize, flower flies, awesome. Maggots, even cooler. Larval flower flies can reveal new scientific insight that, you know, people haven't been giving the credit it deserves, and that community curiosity is pushing science forward. It would take much longer for scientists who kept to themselves, kept in their little silos, to do the work that just everyday people can contribute to, right? So incredibly valuable, and I'm hoping that the realm of entomology and broader environmental science in general can come to appreciate just how important people are. So curiosity leads to trouble, and this is the best kind of trouble, and I've been so grateful to learn what I can, talk to the people who are interested and keen and noticing other things. It's been an incredible project, and I love the work, and I hope you love flower flies now. Okay, thank you. Usually, during the presentation, I like a back and forth. So if, you know, I didn't explain something clearly, or, you know, you thought of a different angle on something that I'd said, like, I really do appreciate that back and forth. But this is a much bigger crowd than what I usually speak to. So now is your chance.
And so, Alice, I have a microphone, so if anyone wants to raise your hand, I can come to you. I'll just ask the first question. You said the term barcoding earlier. Can you explain what that means?
Sure. So barcoding is essentially the process of taking a specimen, an insect specimen, a plant specimen, what have you, treating it so that the DNA can be replicated on both ends, I think, and that pattern, if a pattern already exists within a database, it can match those together, so you get an identification that way. So it really depends on the quality of the barcode that you're submitting, as well as how extensive that database is. If it's a new species or, you know, something quite different, then you're likely to not get a match, and that would be, you know, at least a starting point for developing that argument. But if it matches to something, then you've got at least a name to what you've submitted.
Cool, thank you.
Yeah, it's just how long is the life cycle? Like, how long are they a fly? How long are they a larva? How long are they, you know? Because without knowing that, I might look at one and it might be there one day and not be there the next day.
Yep.
So, explain that a little
No, it's a fantastic question. I'll say the answer is kind of, you know, it won't satisfy you, unfortunately, but it varies. So the larval will stay in their larval stage until they can, they've accumulated enough energy to go through metamorphosis or to go through into their pupal stage. So for some flower flies that are living in, you know, energy poor, you know, prey, like whatever they're feeding on, if it's not of a high quality, they have to spend longer in that life stage in order to accumulate that energy. I think there are a few that the larval stages can be like over a year long. Yeah. So it depends, but it has to do with energy.
Any other questions? Okay. While we're on the subject of larvae, where do they overwinter?
Who rakes their yard? All right, so good.
They're in like dead leaves, basically.
Yes.
Under them there.
Yep. So there are some syrphids that, like I mentioned, will migrate. There are some where in their larval stage, they will go into, what is it called? Like they'll experience less activity, their metabolic rate will go down, and they'll be within the leaf litter because that acts almost like a sweater for them to the cold air and snow and things. So if you're raking up your leaves, you might be moving or harming flower fly larvae unintentionally. Same with many other beneficial insects.
Can you define that delicious term maggot?
Can I define it?
What makes a maggot?
It would have to be an immature stage. You'd have to like confirm that it's the life stage after it comes out of an egg. And for the most part, I think that it's attributed specifically to flies. So you wouldn't necessarily call, you know, a beetle larva a maggot. Maggot is specific to flies.
I'm curious, you talked about rearing them.
Oh, yes.
So, if you find one on the plant, do you just take the whole plant or something? Do you have aphids in the lab that you feed them? How do you ensure survival? How do you do that?
Yes, so I've done this for maybe 3 years now, where I've spent the summer collecting larvae. And I can tell you, at least through my experience, they're not very picky. A lot of them are going to do well in any sterile container that you have. And if you find it on the tip of a plant that is also covered in aphids, and you just, you know, kind of cut the tip of the plant and you put that into your container, you really don't have to do anything else. Make sure that the environment isn't stagnant, like at least there's some airflow, and make sure that, you know, mold isn't developing faster than the larva is. But for the most part, they are just happy to kind of be in their little container, eat the aphids that have nowhere to go, and then pupate.
Okay, so basically my question was about like how you basically identify things like mosquito eggs or like larvae. Basically, that's what I'm asking about here. It's important like topics for me like to find out. Basically, Yeah, no, that's a great question. You know, flies can take on, especially the fly larvae, they can take on a lot of different forms. From what I understand, like the example you gave with mosquitoes, if you look at the phylogenetics, so like how things relate to each other over long periods of time, there is a split within the fly order that separates where some maggots will have that head capsule-like thing, and some won't. So the group that I study, they won't have that head capsule, but things like mosquitoes would have like a more obvious head and eyes. yeah. Thank you.
I was just wondering, when you were looking at potentially identifying a new species, made me wonder, how do you know it's not a hybrid? Do insects hybridize?
Oh, I feel like I've heard I think insects do hybridize. I'm not too familiar within syrphids how often that is. And going to the evidence that I have for this undescribed species, so the adults morphologically, I'm still trying to figure it out if they do have, you know, the tiniest difference that's consistent across our specimens. That's work still has to be done. Thankfully, very quickly, sometimes it takes a very long time to, you know, have a gem like this, but I was dissecting the genitalia, so the internal components, and I found a fairly consistent character of the hypandrium, which is one of the plates that kind of keeps the genitalia together. And in Eupeodes palmis, so the one that it's morphologically closest to, it will have a very smooth margin to it, versus this undescribed species, it consistently has a bump. Like it's this, not elongate, but it's got a bump to it. And the having differences in the in the genitalia can be very important, because you wouldn't expect for opposite sexes of different species with different genitalia to be able to match up properly. Yeah. And then on top of that, if you look at kind of like the softer data, I would say that our particular undescribed species, I have not found it on anything other than a specific type of aphid on a specific type of tree. Whereas the other two, very generalist in their larval forms. I can find them, I don't know, like on oleander aphids, on milkweed. And then I can find them on, oh man, an aphid of a different shrub or a different herbaceous plant. So the strategies that they employ or that their ecology is, you know, I would say different.
Right.
Mm-hmm. And there's also, additional, I still have to look into this, so I won't say that it's like, you know, a for sure thing. But if you look at iNaturalist, if you look at the records of the green larva, which is again kind of our baseline for comparison, and then you look at some of the observations of our undescribed species, they'll overlap in some areas, but there are some spots where they don't overlap. So that's maybe giving you an indication that they're using the habitat differently or have different habitat requirements that would separate them. Thanks for the question.
I can go next. I guess I have 2 comments and 2 questions after that. One is, you can really tell you guys like science outreach and knowledge really came through in your presentation.
Thank you.
So to say, good job for that.
He's over there.
Oh, good job. And that it's nice to see that you really love it. So that's always so nice to see in presentations.
Sorry, I did not know what a flower fly was before 2022. That's how deep and quickly I've fallen down this rabbit hole. Yeah. You can.
And I guess my 2 questions. My first one would be, is there any research, or does anyone look into how the maggots or the flies may be transferred over large distances? So I'm just thinking like how we got ticks in Canada on birds and migration from birds and dropping and then more until winter. So just wondering, just with the one that hasn't been found in Canada previously, like is there any chance that they could be transferred on, say, like food products that maybe haven't been inspected or they're not inspected for that, or if there's anything known about that, or like how birds or
For this Hamilton observation?
Or like any of them, like would that be a possibility?
There have been species that have been introduced. For example, a very common one is Cerida pippins, I think. And that is associated with like decomposing food. Thankfully, there's maybe only one flower fly that exists in North America that you could argue is invasive. For the most part, we're very lucky that they're not disruptive in a critical way. In terms of their movement, I'm not too familiar of cases where, you know, you'd have a southern species that's being transported long distances. But we are seeing that happening naturally with climate shifts. But not a great answer, I'm so sorry. It's possible because there are examples of it, but of native species, I don't know of any cases.
Okay, that's cool. And then just my last one is, I know that there's tons of different species, but how, I guess, scalable would be growing? I don't know if that's the right word. The maggots, like if it was for agricultural, either control or pollination or both?
At the moment, so this is a great question, and it ties into kind of work. The precedent has been set in Europe. They are decades ahead in terms of developing biological control agents of flower fly larvae. They have quite a few options available on the market, or their market, but for biosecurity reasons, we can't just kind of use their fauna and bring that fauna here to do the job we want them to do. So there have been researchers at the University of Quebec, and they're doing a lot of great work on one species of flower fly, and that's Eupeodes americanus. They've gotten it to a point where they were able to produce it at high enough levels and, fairly, have a stable population that they could market that and have a product come out of it. As far as I'm aware, that has they've reduced that. They're no longer offering it as a product. But there are other scientists, even in Ontario, that are looking on expanding to other species. In terms of scalability, it's hard because once you get an operation to a very large size, then you have to consider things like contaminations, different types of pathogens. If you get a single parasitoid wasp in your aphid colony, that's pretty much garbage, and you have to start from scratch. So there are a lot of points in the process that can compromise those large productions of flower fly larvae. But it can be done. It just has to be done in a, very peculiar kind of way.
Hi, Alice, I'm in the same.
Oh, yes, hi.
I'm Sheila. I just want to first of all, compliment you on your ability to speak to lay people who learned about fireflies. Flower flies.
It happens to me too.
When we saw the upcoming meeting, that's how long I've known about them. I just know much more than I did until tonight. But I'd like to be a citizen scientist. So if you were giving us quick directions, because we were all going out tonight, this morning.
Okay, yeah.
To look for specimens for you, what would be the directions that you would give to a team of us if you were just arriving at the sanctuary and our goal was to try and support your research?
Okay. Are you hoping to get more of like the adults? Because that's probably going to be the easiest. If you're looking to just start almost like a species list, let's say for a particular property, the adults are going to be easiest to start with. So go towards things that are blooming, and go in kind of I wouldn't say early morning, but between, 9 o'clock and 12 o'clock, just before that, hot part of the day hits. That's when many syrphids, not all of them, but many of the flower flies are going to be looking to get their floral resources. So if you're at the flower when they also want to be at the flower, then your chances of finding, different species, especially if you're in different habitats, that's probably going to be your best shot for diversity. And then if it's larvae, the larvae are tricky. You have to have a good sense of when a plant looks sick or like when a plant looks off, which gardeners might be very familiar with. You can kind of see from a distance if a milkweed, for example, has a bunch of orange little spots on it, right? It takes some practice, but once you get it in your mind of what the plant should look like, if it has those really big colonies that form almost these like irregular shapes or these like little bubbles and blemishes, then you'll pick up on that cue to maybe check it for larvae. Great question. Please go out. Everyone get a net.
Thank you. I have two questions. So how useful have identification guides been to your work? What should the last few slides?
Yep.
The last slides shows that the idea is to be genus, level of genus.
Oh, this one here, right?
Yeah.
Yeah.
How helpful have these been in general to your work?
For me, even sorry.
Follow up with the other question.
Yeah, ask the next question.
So the other one has to do with the new species you found. So I'm wondering, there are many ways new species are made, right? So it could be geographic isolation.
Yep.
Right? But also there could be species located in one region that just don't reproduce anymore, right? And then form some new species.
Sure.
I was wondering, how did you arrive at the hypothesis that you're dealing with a new species? Is it just the morphology being different? Did you test out if it successfully or unsuccessfully reproduces with the other known species?
Yeah. So great question. I'll answer the last one first. So I love that you specified that a undescribed species or a new species, it's a hypothesis, which is a fancy way of saying like you need to have an argument to make your case. So thank you for that. Not a lot of people get that nuance. And I'd say that you're right about species sometimes coming about by way of population separation. And in this particular case of the undescribed species, the evidence there, as far as I'm aware, is not, we don't have any evidence that they hybridize. When you look at the genetics, it's within 1%, it's within 2% of Eupeotes americanus, but there were actually existing barcodes. There was one existing barcode from Alberta of a specimen, and it was an exact match to that. So what that tells us is that that particular barcode reading, or that what the barcode spat out, it wasn't an accident, right? It was actually replicated, or, you know, there was another specimen that was exactly the same as it. So that's one piece of evidence genetically. Like I mentioned, there's the ecology to it. We find the other two specimens on plants and aphids very broadly. Their range is quite broad, whereas this particular species, it's one aphid, one plant, and as far as I can tell, or at least based on my experiences so far, I've only been able to collect the larval stages in June. So they have a very restricted window where they're in their larval stage, as far as I can tell. But of course, I'll need access to specimens from across Canada, right? Because like I mentioned, it's all the way out in BC. I'll need access to those to confirm, like if one was found in August or one was, I don't know, popped up in May, those kinds of things. Other evidence, yes, the morphology. I'm still looking into external characters. I only have maybe 12 specimens, maybe less than 12 specimens. So if I'm going to be making a strong argument that it's this particular feature, you know, it's this hair on this plate in this part of the body on the females or on the males, you need an extensive reference collection. So still working on that. Hopefully something comes up that adds to that story. Yeah, I think that's what I have for you that argues that it's a new species. Nothing about hybridization, unfortunately, as far as I can tell, but we can look into it for sure. And then the first question had to do with this, right?
Yes.
And you mentioned that it's at the genus level and how that's helpful to the work.
Yeah, in another of the slides you have, there is another ID group.
Oh, like the references?
Yeah, so I was wondering how useful in general have these groups been to you in ID species?
Mm-hmm.
Yeah, it's key to general, right? It's not key to species.
Yes. So I'm not always familiar with the other subfamilies. I focus very much on Surfinae. So it's the predaceous, really colorful ones. But Aristallinae, there are lots of species in that. Some of them are very difficult. And early on, I was using these just to kind of know where to look in the book instead of flipping each and every single page until I found something that maybe worked. So it's a really great starting point for establishing like what you're looking at, the characters that you might consider in order to get it to a certain place or to a certain identification, and then familiarizing yourself with kind of the smaller details in something like the Manual of Nearctic Diptera or like the guide, like this one. Yeah, so I would say that even at a genus level, I believe there are papers that discuss that species level can be helpful, but so can genus level for ecological studies, and it all depends on the type of scientific question you have. So if your question is something like, what kind of flower flies are in an agricultural setting versus in an urban park, and you're trying to make conclusions based on what you know about the biology, you might not have to go down to species level because Sometimes they'll be very different, but a lot of the times they're going to be very similar to their sister species within that genus. Is that good? Okay.
We've got time for a few more questions.
Yes, you said that the flower flies are quite resilient. How are they with pesticides?
Oh, that's a great question. I will say there has to be a lot of nuance when responding to questions about pesticides and their potential impact on like any single organism. It really depends what the pesticide is, when it's being applied in the time of year, what life stage you're capturing it at. So for example, flower fly larva, if it's on a infested plant and it gets sprayed directly or passively with pesticides, it's the food that it consumes will have pesticides on it. The surface that it's navigating will have pesticides on it. And they're going to spend their whole life in a very small area that has been treated, right? So in those cases, they're probably not going to have a great time. The pesticide is going to do what the pesticide is going to do. Versus things like adult flower flies, if they get sprayed, their sensory cues kind of get out of whack, or for some reason they make the decision to leave, they can, and limit that exposure as they go into a different habitat. So yeah, it's, I don't know of any scientific papers or academic papers that look at the rate of pesticide or specific pesticides, but I would say that I would guess that they're aligned with how other flies respond. Mm-hmm.
Yeah, I was just wondering about the second part of a question asked earlier. How long do the adults live, and what kind of life do they live? Are they plant specific?
Mm-hmm.
Do they reproduce just once and die?
Yeah, that can vary as well. You can have flower fly adults that are multi-generational, so they will be able to mate and have offspring, and then those offspring mate and have offspring, and it continues for the length of their active season. So some of them multi-generational, some of them just one generation a year. I'm trying to recall if there are adults that kind of go over 1 year. Nothing's coming to mind, but there might be. I won't say no, which is kind of cool to think about. Sorry, what was the next part of your?
How long do they live, basically?
Yes, so how long they live, it's, I think, going to depend on the species.
What do they eat?
The adults?
What do they eat?
Basically, most of the adults are going to be feeding off of nectar and pollen. Some of them are very specific in terms of their plant host. They have, some of them, and I would say that it's not often the case, but for example, there is a flower fly, I believe, on the West Coast, and that has been kind of found to visit a very specific type of orchid, and primarily that, right? And so we see instances where there are these very profound, very interconnected relationships, but for the most part, I think flower flies are generalists in that they'll be happy to feed on whatever plants are blooming at the time, and they do show a preference for kind of shallow flowers, because they don't have those long hummingbird tongues or anything. They need to access the nectar, as well as, I think, white, yellow kind of flowers, and if the plant has like an umbel shape to the blooms, because it's just easy.
To echo what a few folks have said tonight, your knowledge, your passion is amazing. What is next for you in the next couple of years, and what can we see you presenting on back here in a couple of years?
Oh man, if I could dedicate my whole life to this, that would be incredible. Wow, that would be a dream come true. Like I mentioned, I was introduced to flower flies in 2022, and I was doing it as a hobby for maybe the first year and a half. And my intention was to continue that hobby for the rest of my life. I think flower flies are so interesting. There's so many different things to learn about them. So when this PhD project was brought up, or I was in conversation with that, I was like, I get to do this for 4 years and someone pays me? That's insane. So if I could continue doing research on flower flies in a way that can be translated to just like, it being useful to people, I think that would be my ideal. But ultimately, like I mentioned at the beginning, like all insects are so valuable. While flower flies will have a special place in my heart, it wouldn't be terrible to focus on something else. Yeah.
Well, this has been such an amazing crossover of nerdiness and really cool at the same time. So thank you so much on behalf of the Hamilton Naturalist Club. Let's give Alex a round of applause. Thank you so much for being here tonight. Go look for flower flies tomorrow and submit what you can to INAT. Maybe we'll see you back here next week for the birth study group meeting or in a month at the AGM. Regardless, thank you so much for coming. There's still cookies available. So keep mingling if you would like. Otherwise, have a great night, everybody.
I do have like a larval specimen encased in resin if anyone wants to see it. Like actually hold it in your hand. Yeah. So this is something I made myself.
Yeah. Wow.
Whoa, right?
Yes.
Yeah.
I think the short percent green venture is that works at the Arboretum as like well, like, and I know like